and a warm welcome to today's talk. We're going to be going over the second part of our Genetics of Neuroanatomy lecture today. So it's kind of more of the same, but an extension on what we've looked at previously. So without any delay, let's jump in. So here's the title slide. Welcome back, everyone, by the way, after the Thanksgiving holiday weekend. I hope you all had a good time. I hope it was restful and relaxing. I know that's kind of like probably not possible, at some level if you're busy students or busy with jobs or life in many ways. Having a day off class is not necessarily going to be, you might need a little bit more than that. But I guess fall break is coming up, so hopefully you all can stretch out and relax, and I can catch up on making more lectures for this class. So just a few elements of course business to discuss. First, we have the midterms have been graded for the most part. They'll be available, or the grades will be available on Brightspace once everyone's, once all the grades have been sort of finalized. And I'll talk about the grades in the midterm in a moment. The second assignment is also now available if you haven't noticed. So it's the same kind of concept as the first assignment. It's a sort of web quest or a web-based exercise where you watch me explain a few different concepts, and then you go in and you follow the directions in the assignment and produce the kind of data and analysis that you need to, and learn a little bit about a certain kind of bioinformatic, a certain set of bioinformatic approaches to data. And so, that's due, I think November 8th or something like that. So please check on that and take a look at that and get started as soon as you can. As always, it's better to do it sooner rather than later. Okay, so let's go into our review of the quiz results. So, here is the distribution of grades. So this is a histogram with the density, kernel density function overlaid here. So the class average was 72.83%, and the standard deviation was 14.91. I did actually apply a bit of a curving to the grades to just kind of give them a little goose in the right direction, just to make sure the average was a little bit better. That's fine, though. It was a very difficult test. I can certainly see that. So I think actually everyone did quite well considering, and it should hopefully be a good flavor for things to come. So your individual grades will reflect kind of the grades that have been computed here, and then you'll be able to go and and see what you got soon. So I also, so here's the class average, and this is your kind of typical thing. And I wanted to ask a couple of other questions too of the data, given that it's available, and maybe you'd be finding this interesting or not. So one thing I wanted to ask is the score that students received a function of how long it took them to write the exam? You might think that students who spend the longest amount of time writing the exam might get better marks. What's surprising actually is that almost, probably the majority of students took the maximum amount of time possible. So in this plot, we have the time to complete as a percent of the allowed time. So if you used 100%, then you would have used your full 90 minutes or your full whatever number of minutes you were given to write the exam, depending on your accommodation status. So most students really took the whole time. And that wasn't exactly, there's a sort of a weak negative relationship there, so that the longer, on average, the longer you took to write the worse you tended to do. But I wouldn't really consider this to be a real effect. It seems to be just sort of artifactual based on the fact that most people took the full amount of time, and there's a wide range of grades there. And then another thing that I thought would be funny to ask would be, does the amount of time it takes you, or does the amount that you delay the start of the exam have any relationship to your score? So you could start the exam at time point 0, meaning you write at 8 a.m. on Monday morning or whatever day it is, or you could start at 24 hours after, which means you write at the just before 8 a.m. the next day. So So that's kind of an interesting question, because you might think that, well, if students wait till the end to write the exam, that would give them almost 24 hours extra to study, and that could be an advantage. But it seems as though it's not really. It's a bit of a negative relationship. So the longer students waited to write the exam, the worse they tended to do. Although, like I said, the variation is quite wide here, so you can't make any serious connections. I guess you would if you were looking at these data and you saw that the grades got way better for students who waited to the end, you might wonder if people are sharing their answers, or there's kind of communication going on. So I guess that's something to look for, but I don't think any of you would do such a thing. And then so finally I mapped this out on a heat map and applied some clustering, as we love to do here. So these are the questions that had written components. You can't really cluster the multiple choice because you can either get a 0 or a 1, and there's no other grade possible. But here the color scale of this heat map is the percent of grade, the grade percent for each question. And the question numbers are given here in the columns. And then I applied a k-means method to split the rows into 2 clusters. So you can kind of see here, each row is a student. There's kind of 2 separate clusters of students. There are students who did fairly well uniformly for all of the questions. And then there's a cluster of students who had a lot of difficulty with questions 7, 9, and 14. So by that kind of standard, those 3 questions would be the ones that sort of make or break your performance on the exam, and would do the best job of discriminating between people who get high scores and low scores in the exam. So I think this is kind of interesting from a quantitative point of view, but it also points us toward maybe the questions that are the most important to review. So I'm going to take a few minutes now, while you're all with me, to go over those 3 questions that seem to give people the most problems. And just kind of review what we were looking for, and maybe you can reflect on what you remember writing, I guess, and think about whether that works or not. So first we have question 7, and so this question asks, genetic variation can affect protein coding sequences, but this is relatively rare. Remember, exon SNPs that are in exons are comparatively rare compared to SNPs that are somewhere else. SNPs are much more common in non-coding regions of the genome. Explain 2 mechanisms by which non-coding SNPs can affect the molecular biology of the cell. 4 points. Explain the importance of tissue and temporal specificity in discovering these effects. So what I was looking for here was a description of EQTLs and MQTLs, which you could find on lecture 2, slides 12 through 16. So there, what I'm kind of getting at is that if you're not going to affect the protein sequence, but you're still going to affect the molecular biology of the cell, the main thing you're going to be thinking about is either epigenetics or gene expression, or some combination thereof. And so genetic variations that have an effect on gene expression are called EQTLs. We can look at how the expression of a given gene in a given tissue is influenced by your genotype. And then MQTLs, which are methylation quantitative trait loci. Those are situations where the level of methylation in a given CBG site or at a given locus is influenced by your genotype. So those are the 2 mechanisms that I wanted you to describe. And this could include, although it wasn't necessarily specified, descriptions of how transcription factor binding site mutations can lead to changes in molecular biology. Really, TFBS mutations, transcription factor binding site mutations, would kind of relate closely enough to EQTLs and a little bit to MQTLs, because ultimately transcription factors, what they're doing is binding with the DNA and influencing gene expression. So if you have a mutation at a binding site for a transcription factor, either at a gene promoter or an enhancer, how it will manifest itself in phenotype, at least at the level of the cell's molecular biology, would hopefully be, or would be expected to be, a change in expression. And then as I explained in those slides, because gene expression and epigenetics are tissue specific, you have to look at in the right place in the right time. You can't just take any old tissue. It's not like DNA. DNA is the same in every cell, so you can take any tissue you like and measure somebody's genotype. To measure their gene expression and their epigenotype, you need to look at the tissue that you think is relevant. So that's kind of a lesson from the GTEx study, where they were looking at EQTLs in all different tissues. So that's what I was looking for there. Number 9 here, most gene expression is biallelic, but this is not universal. Some genes are expressed monoallelically. What is the difference between biallelic and monoallelic expression? We discussed 2 examples of monoallelic expression in class. Explain each example, what purpose we think it serves, and describe the role of DNA methylation in maintaining monoallelic expression. So I was kind of proud of this question because it requires a bit of synthesis across separate lectures. So this is something to look out for when you're studying for your next exam and for your final, is that these are the kind of things that I would be looking to ask, is like, this is not one set of slides from one lecture. It's kind of 2 separate lectures describing 2 different aspects of, or 2 different varieties of monoallelic expression. So what I was looking for here is that, a description that would go into, how biallelic expression is when you express genes from both your from both your maternal and your paternal alleles, and they're expressed in roughly equal quantities. And then monoallelic is when one, mono is one, one allele or the other is expressed, or you could, another way to phrase it would be one or the other allele is silenced. So there's kind of a couple of ways that this can happen. I mean, there's many ways that this can happen, but the 2 that we talked about were X inactivation. Females or people with 2 X chromosomes get 1 from each parent, of course, and X inactivation ensures that they only express one or the other in every cell. So this would be an example of monoallelic expression, because you can only express maternal, your maternal X chromosome or your paternal X chromosome, one of the 2. How it's determined is a little bit random, as we talked about in the X inactivation part, but that's an example of a monoallelic expression mode. So you can look at lecture 5, slides 11 through 13 there. And then imprinted genes are the other example that I gave, and that's lecture 6, slides 20 through 30. Imprinted genes are not necessarily on the X chromosome. They could be on any autosome, but they do have this unique property where you express either your mom's copy or your dad's copy. The copy of the gene that's silenced is called the imprinted allele, and then the other one is called the expressed allele. So you'd want to give those, and then you just go into a brief description of how they work. The way that with X activation you have exist. This is a long non-coding RNA produced by the silent X chromosome, or the inactive X chromosome, that kind of coats the chromosome and attracts DNA methyltransferases, adding methylation to the chromosome, and then ultimately condensing it down into a little shriveled up bar body, and silencing all the genes there. And then for imprinted genes, you have methylation at imprinting control regions, ICRs, and the methylation state there is allele specific. It's determined early on in embryonic development. It's really more just that methylation there is not erased like it is in most places, and the presence of methylation at an ICR silences the imprinted allele. So that's what I'd be looking for there. People, I think people, many students, I guess, got sort of 1 or 2 of these, or one or the other of these, but maybe didn't make the connection that these are 2 separate concepts, or 2 separate phenomena from 2 separate lectures that both are examples of monoallelic expression. It was kind of a little bit between the lines. So I'm kind of, happy with that question in the sense that it required a little bit of abstract thought. Not really abstract thought, but it required you to kind of extend your thinking beyond what was explicitly told to you. And I think that's probably a good way to make questions, is not to, is to try to make you think on your feet a little bit and put things together. But it also naturally, I guess, turned out to be one of the harder questions. And then, so here's number 14. This is the third question that we had sort of the lowest grades on. And here it says, of all the epigenetic marks we discussed, DNA methylation at CpG dinucleotides is the only one with a known mechanism for mitotic inheritance. How does this work? And what is special about CpGs in this regard as opposed to CPAs or CPTs or CPCs or CP anything really? CPH would be another kind of CP something. So here the answer is... or the way that mitotic inheritance of DNA methylation works is that when DNA is being copied, what you end up with is what's called hemimethylated DNA, where one strand has preserved the methylation mark that was on the DNA in the first place. You kind of have methylation marks on both strands. They split up. Then each daughter strand, or each strand has the methylation marks that were already there. And then as DNA polymerase comes in and fills in the blanks, you have DNMT1, which is the maintenance methyltransferase, coming in and copying the methylation over from the hemimethylated, hemi meaning like half methylated, and making it fully methylated, or fully methylated. And so because of this, cell identity, which is what DNA methylation does a lot to control, is able to be perpetuated across generations. this ability for DNMT1 to fill in hemimethylated DNA. So this is from lecture 5, slide 8, if you are curious. And the reason, or what I wanted you to say, was that CPGs are special because they're palindromic. So a CPG on one strand guarantees that there'll be a CPG in the same place on the opposite strand. This isn't true for all the other ones, right? Because G is complementary to C and C is complementary to G. So if you have a CG over here, what you'll have on the other side is a GC as a reverse complement. So CPGs are the only place where this is guaranteed. If you have a CPA, the complement to it would be like a GT or something. So you're not going to even have a cytosine on the other side of the DNA to methylate. So only CPGs really have this property of being able to perpetuate DNA methylation across mitotic inheritance. So that's what I was looking for there. A lot of people did get this, and I was happy to see that palindromic, the word palindromic was mentioned quite a lot. Some people, I think, what I saw was we're kind of getting into things about CPG islands and other kinds of things that aren't really relevant. CPG islands are a place where there's a lot of CPGs. This is true, but it has nothing to do with the question about how sort of the molecular biology of inheritance of DNA methylation across mitosis. So that's 3 questions here. So your TAs are going to be in contact with you whenever they work this out, hopefully, soon within this week, to arrange kind of a schedule for viewing your midterm and going over the questions and the grades in a little bit more detail if you like. So if you want to hear more about this, you can do that. Or, I'll be holding office hours this Thursday. You could also come visit me if you want to hear my take on things. I don't really want to spend the office hours going over your specific exam. That's something you should do with your TAs. But if you just have questions about the content, or if you want me to just kind of riff on the material and clarify anything that you think you had trouble with, then by all means, stop by and visit. So I think, so I think the grades here were a pretty good starting point for everyone. It's a lot easier to have a lower grade on the first midterm, and then have it go up in subsequent midterms. It's a lot better to have that situation than to have a really good grade on your first midterm and then have it go down. So I would stay optimistic. If you're not getting the grade you were hoping for, hopefully at least you learned about the kinds of questions I like to ask and the kind of things I want you to focus on when you're studying, and that'll hopefully guide you. There's a lot more points to be earned in this class, so please do not fret if you're not getting the grade you really want to get yet. It's very early days still. There's lots of assignments and lots of more tests to come. And I guess another sort of thing that I would say, this is a third year class, and I guess I sort of like to think about how all of you, or many of you, would have only been in high school a few years ago, and however well or poorly you did on the exam, could you imagine even having a remote possibility of even getting one question right in high school? Imagine doing any of this when you're in high school. It's you really have to kind of sit back and appreciate how much you've learned over the few years you've been in university, and how this material, which would be incomprehensible, I would assume, to the average student in first year even, now you're all doing fairly well with it and handling it handling it with a lot more ease than you ever thought you could. So even, like I say, even if you're not, super happy with your grade, you really should sort of take a step back and appreciate the intellectual progress you've all made. It really is quite impressive. And the other thing, by the way, is that you should never stop learning, because the amount of kind of change you've made year to year, you're in your third or fourth year maybe right now, you can continue making those kinds of leaps every year of your life going forward. You don't have to be in university to learn. You can spend your own free time learning as well. And so, it's just kind of nice to keep watching your improvement on things and enjoying the progress. So I do have one question here. So do you have any tips for studying, as I thought that I was a lot more prepared than I ended up being? Yeah, this is always the problem, isn't it? When it comes to exam preparation, is I think there's an element of sort of maybe psychology in understanding your teacher or your instructor or your professor, and thinking about the types of questions they would be likely to ask. Maybe this exam has given you a sense of where I'm at in that regard, and will give you a better sense of of what you want to focus on. Like I said, I think what I like to do is kind of make connections across multiple lectures. So certain things are going to come up over and over again in lectures, things like the importance of tissue and cell type specificity, the importance of clustering and machine learning methods in making interpretation or interpreting genome scale data, the importance of linkage in genome-wide studies. You'll see more and more of these coming up. And so what I would want to do, or what I'd want to counsel you to do, is try to look across lectures and pick out common themes and common concepts, and imagine me asking you a question like that. The way I used to study back in the days when I was taking tests was I would try to go through the material and invent as many questions as I could think of if I were the one creating the quiz. And then I would go through and I'd try to answer them. And that turned out to be a fairly good system because making questions is hard. There's not that many things that would really make it good questions, especially when it comes to written long answers, because I can't, it has to be long enough that you actually have room to explain it, and it has to be abstract enough that you couldn't just open up the slide and kind of copy the material off of it. So you kind of have to get into psychology a little bit to know where to focus your studies. That would be my best piece of advice. I was thinking actually when I was just looking at the ridiculous amounts of time I used to have to devote to studying, I was looking at these like flashcards I made when I was an undergrad. I was digging through my house and just looking through old material. This would have been from like 12 years ago or something, but I used to just try to study anatomy, and I used to design all these like diagrams and map out all the different structures and muscles and parts, and it'll have all the things listed on the back. So if you have an enormous amount of time to devote to things, you can make some pretty detailed instructional material. And if you can add in sort of a bit of psychology about your professor and the kind of questions that they might think of, then you're going to be ahead of the game. So hopefully that will be helpful to everyone here. Like I said, other than giving these lectures, my only other job really is to try to help you learn things. So if you feel like you need more help learning, that's what I'm here for. So let's get into the material now. So we're talking about the genetics of neuroanatomy, and focusing now on kind of a continuation of the single cell stuff we were getting into last class, with another expansion on just the sort of ridiculous high throughput methodology that are used commonly these days. So here we're looking at what's called spatial transcriptomics. So single-cell RNA-seq, which we talked about last time, was a major advance. It allows you to basically do kind of a high throughput classification of cell types on the basis of their gene expression patterns. However, the anatomical resolution is rather low, because ultimately what you're doing is micro-dissecting out a certain part of the brain that you're interested in, and kind of separating all the cells and doing the sequencing on them. And some dissections are very relevant to people's curiosities about anatomy, but very difficult. For example, if you're trying to separate the dentate gyrus from the hippocampus, that's quite challenging. Or if you're trying to look at the claustrum is a thin band of gray matter. It's medial to the insula, but lateral to the internal capsule. Very, very, very thin slice of the human brain. Tough to microdissect that. So there's another method that sort of addresses this called spatial transcriptomics. And what's happening here is you're taking slices of brain, as you would if you were mounting them on a microscope slide and looking at them with, a Nissl stain or something like that, and putting them on these special slides that have thousands of microscopic spots. And each of these spots is positioned in a grid on the slide, and it's coated with, well, let's just call them various chemicals that will capture the RNA exactly in that spot and then save it for sequencing. So each spot is uniquely tagged with sort of a DNA sequence-based barcode, and then the chemicals on the spots enable all of the RNA that is directly over the spot to be captured. So what you end up doing is basically capturing all of the RNA, say, that's in this spot, all of the RNA that's in this spot, and labeling it for RNA sequencing. So now you have an RNA sequencing reaction where you're doing, I don't even know how many of these are. There's a lot. There's like 7 times 7, whatever that number is. So there's, hundreds at least of spots here that you would get separate data from. And because these spots are located in a kind of known, have a known location, you'll be able to get all of the gene expression information from this spot, all of it from this spot. And they kind of act like pixels in a photograph in a sense. So Now you can do this to map your gene expression data onto the microscopic anatomy of different brain regions. So you'll have RNA-seq data that's genome-wide, and you'll have it localized to very, very tiny, a whole grid of very, very tiny sections of the brain that you're looking at. So I'll give an example of how this is used in sort of the neuroanatomical concept or context by citing a paper by Maynard and company and published recently in 2021. And here they're looking at the neocortex. Now, if you recall from your other classes, the neocortex is a layered structure. It has 6 layers. And we can't get into the layer functions here, but there's different things going on in every layer. There's different cell types. Each layer receives inputs from different places and sends outputs to other places. And there's a whole kind of complexity to the layered structure that's been known for quite a while. The most superficial layer is layer 1, and the deepest layer is layer 6, and then after that you have the subcortical white matter. So what they're doing here is using using spatial transcriptomics to measure gene expression in slices of the human cortex. So they have sections of the dorsolateral prefrontal cortex, Brodmann's area 46, and they're mounting them on these special spatial transcriptomic slides like this, and then they're measuring gene expression in all of the different pixels of this slide. Every pixel, remember, is a separate RNA sequencing reaction. So for every pixel on this slide, you have data on thousands and thousands of genes. So just as an example of what you get with this, I'll show figures from the paper. So the slice is this here. This is what the slice looks like under a regular microscope. It's been stained with a Nissl stain, so you can see the cells and the white matter and whatnot. And then overlaid is a heat map representing the level of expression of a certain gene. So here the gene is SNAP25. The red points are high expression and the blue points are low expression. And you can see there's low expression here in the white matter, WM, and then there's high expression all through the cortical gray matter. SNAP25 is a marker of neurons. So you see high expression where there's a lot of neurons, which is the gray matter, and then you see low expression where there's not many neurons, which is the white matter. MOBP, on the other hand, is a marker of oligodendrocytes. Those are the myelin-producing glia. That is expressed in high levels in the white matter, but it's expressed at very low levels anywhere else. And then PCP4, this gene here, you can see it's expressed uniquely or mostly by layer 5 neurons that live here in layer 5. And you can see that the expression of this gene forms kind of a band that corresponds to layer 5. There's some light amounts of expression elsewhere, but the strongest band is concentrated in layer 5. So here they're just showing that they can kind of, using just gene expression data from this spatial transcriptomics approach, they're able to reconstitute the known anatomy of the cortex, something that you would have previously only been able to do by looking at the cells under a microscope and, are these granular cells or are they pyramidal neurons or whatever else. So that's pretty cool. And now there were these kind of known variations in gene expression across the layers of the cortex. That was not some kind of big secret that they uncovered in 2021. That has been known for many, many years. However, it's only known for a handful of really well-studied marker genes. There are 20,000 some odd genes in the human genome. We don't know the layer specificity of all of them. We know that some of them are only in neurons, and we know that some of them are only in glia, and we know that some of them are, for example, enriched in layer 5. But most genes, we have no idea where they're enriched, if they're enriched in any particular layer at all. So the advantage to this study is they can make new discoveries about where different genes are enriched in the human cortex. So here are some examples that they gave. That they discovered. AQP4, aquaporin-4, that's a water channel. It's a channel that allows water to come in and out of cells, is enriched in layer 1 of the cortex, and it also has fairly high expression in the white matter. TRAB-D2A, which is a metalloprotease, whatever that means, is enriched in layer 5. You can see it's following that same kind of pattern as PCP4. So you can see the expression of it is enriched in layer 5 compared to the other layers. That's what this box plot is showing, by the way, is just like the expression of these genes in the different anatomical layers. HPCal1 is enriched in layer 2, and so on. And then, so this is how they determined it using the spatial transcriptomics approach. And then after they went through the entire genome and had a kind of catalog of new layer-specific gene expression discoveries, they could come in and use in situ hybridization, which is a technique that measures gene expression in slices of tissue using complementary RNA probes. They were able to show that yes, aquaporin 4 really is expressed in layer 1 of the cortex. This is a kind of a novel discovery, and the only way to discover it, well, you could either discover it this way, or what you could do is just go through and stain the brain with 20,000 different probes and look at them one at a time and see which is located where. So the method that they've got is certainly a lot more efficient. So this is a new level of understanding of the layered structure of the cortex that would have really been very difficult to determine without these kind of spatial transcriptomic approaches. But what good is it? Well, a common sort of theme that you'll see, I guess you might call this a hint. Well, I shouldn't say it's a hint because I don't have any idea if I'm going to ask this, but this is the kind of thing I'm talking about. Like, the idea of trying to take epigenetic or morphological or take one set of data and try to link GWAS hits onto it as a way of understanding how genome-wide association studies actually work is a common theme. We'll get into that fairly often. So here, what we have is a heat map where they're looking at the association between genes, or between SNPs rather, genes associated with SNPs, I should say, linked to ASD, autism spectrum disorders, as well as schizophrenia. They're looking at the genes that are associated with those diseases and what layer of the cortex they are expressed in, because those are both kind of considered to be neurological or brain-associated conditions. And the question still is, where in the brain are they active? The genes that cause or are linked to ASD, where in the brain are they expressed? That's pretty relevant if you want to understand the neuroscience of ASD. If you are looking in the wrong part of the brain, you could spend a lot of time chasing phantoms. So in this plot, you have a heat map where the different columns just represent sets of genes from different studies. So DE up are genes that are differentially expressed in ASD brains that are upregulated compared to control genes, and DE down are genes that are differentially expressed and downregulated in ASD brains compared to control brains. The layers of the cortex are given in the rows, layer 1 through 6, and then white matter. Within each row, there's a color that indicates the p-value of the association between the genes in that cell and the genes that are enriched in each particular cortical layer. So the higher number, it's the negative log 10 p-scale, as I say here. So the higher number means a more significant p-value. And then for ones that are significant, they have a number here indicating the enrichment. So and you can learn about enrichment when you do assignment 2, but the essentially the amount of times that genes appear here compared to what you'd expect given a null distribution. So what this shows is that genes that are upregulated in ASD brains are 5-fold enriched among layer 1 cortical neurons, as well as 2.5-fold enriched among the white matter. And genes that are downregulated by ASD, or in brains of people with ASD, are enriched in layer 3, 4, 5, and 6. So kind of a way to kind of boil this down is to say, well, we knew that ASD had something to do with the brain. We knew it probably had something to do with the neocortex because it involves a lot of higher level cognitive functions. Within the neocortex, we probably would have guessed that it has something to do with the gray matter because that's where the neurons live. Those are all sort of obvious, well-known points. But what layer of the neocortex is most likely to be involved? Is it the pyramidal layer, or is it the granule cell layer? Like, what layer are we talking about? Well, now we have an answer for this, because the genes that are differentially expressed in ASD brains tend to be in layer 1, or at least upregulated tend to be in layer 1 in the white matter, and then the ones that are downregulated tend to be in layers 3, 4, and 5, and 6. So that's just a simple way of taking GWAS data or any data you have that you already have, know about genes that are related to ASD, and linking them onto specific cortical layers, showing you how you can use these spatial transcriptomic methods to get a much higher level of anatomical specificity and make much more sense of the GWAS data and the various other kinds of data that we're getting about these diseases. And so here, Same kind of thing happening with schizophrenia. So I won't go into it, but you can look at that as well. It seems as though differentially upregulated genes in the brains of people with schizophrenia tend to be in layers 1 through 3. Downregulated genes tend to be in layers 5 through the white matter. And like I said, if you study the brain and know what's going on in these layers, you can start to make up a more complicated story. I'm just showing you this is sort of a way of the genomics data can point people where to look. Okay, so that's kind of what I wanted to say about how GWAS, or about how the modern techniques of genomics can link GWAS data to histology and make sense of the microscopic structure of the brain. But what about gross neuroanatomy? We're talking now about the kind of size and shape of different brain regions. So it turns out that there are structural differences in the human brain that can be attributed to genetics. Now, it's long been a fascination of neuroscientists or neuroanatomists to just measure the size of different parts of the brain. This, I think, is rooted in the fact that people believe that the brain is what makes you smart, and therefore the bigger your brain, or the bigger a certain part of your brain, maybe the more intelligent you might happen to be. This has been a long-standing fascination, and it's not really it's had its successes and its failures, let's just say. But so now that we're in the genomics area, we don't have to break out the calipers anymore and measure people's skull thicknesses. We can use MRI scans combined with GWAS studies to actually see if there are genetic determinants or genetic correlates of brain region size. So the authors here in 2019 did a genome-wide study of subcortical brain structures in 38,851 individuals. This is a gigantic study. They had MRI data on these people, and they also had genome-wide data for people across a wide range of racial and ethnic backgrounds. And their analysis was adjusted for things like age, sex, and intracranial volume and psychiatric diagnosis. So they're not exactly looking at the absolute size of the brain. They're kind of looking at the relative size of different subcortical parts of the brain. And they're trying to link that to genetics. So before I get into this, I want to take a little diversion to tell you a little, to kind of review or tell you about heritability and the way it can be described. The relative contribution that genetics makes to a phenotype can be described as heritability. Of course, this we kind of already have been over this. This is something that might be simpler. Things that are described as genetic can be described as heritable, but not all genetic changes are strictly heritable for various reasons. Broad sense heritability, which is given by capital H squared, or h to the power of 2, encompasses all of the genetic contributions to a phenotype. So everything that is linked to a phenotype that has anything to do with your DNA whatsoever is captured under broad sense heritability. That's why it's called broad sense. It captures every aspect. So this would include additive genetic effects, but it also includes dominance effects and epistatic effects, which we'll talk about. Narrow sense heritability, which is lowercase h squared, on the other hand, it only includes additive effects. And this is sort of partly because the narrow sense heritability is much more simple to measure. Epistasis and dominance effects are quite challenging to measure in a genome-wide way, but also because dominance effects don't typically get transmitted because typically they're segregated upon mating. You might be homozygous or heterozygous or whatever for a dominant allele, but the odds are pretty good that whoever you're mating with may not be. And so after a number of generations, the odds of being transmitted sort of go down and down. And epistatic effects are also not transmissible because of independent assortment and recombination. So epistatic effects refer to a gene or an allele in one place on a chromosome influencing the phenotype in combination with an allele somewhere far away. And these things are not linked, sort of by definition. Things that are far away in the genome don't tend to be inherited together all that often. They can be, but it's not as common. And so they don't tend to be heritable in a long, kind of predictable way. So narrow sense heritability really does a better job of capturing the type of genetic variation that we actually pass on to our offspring. So let me just go over what I mean by additive, dominant, and epistatic effects. So additive effects are the kinds that I was sort of getting at when I was telling you about how odds ratios and allele counting works in GWAS studies. This is a simple thing where a number of copies of an allele you have has a fairly direct relationship with the phenotype. So imagine, for example, you're looking at a variant where if you have the A allele, the capital A allele, you're 2 centimeters taller on average. And if you have the lowercase, you're 1 centimeter, it adds 1 centimeter to your height versus 2, I guess. So if you draw your favorite sort of Punnett square illustration, there's kind of 4 possible genotypes you can have, really 3 technically. You could be homozygous for AA, in which case you'd gain 4 centimeters. You could be homozygous for lowercase AA and gain 2 centimeters, 1 from each allele. Or you could be heterozygous and gain 3 centimeters, 1 from each allele. And this is an additive effect because each copy of the capital A allele you have adds another 2 centimeters to your height. Dominance alleles dominance effects don't happen this way. If you imagine that the A allele is a dominant allele causing brown eyes, whether you have whether you have 2 copies or 1, you're going to end up with brown eyes. And only if you have 0 copies of the capital A allele will you have some other color of eyes. So that's a dominance effect. And then epistasis, which comes from a Greek word meaning standing upon, because I guess one gene's effects kind of stand upon the other gene's effects, and they sort of act in a combination. Occur when one allele has an effect on the phenotype only in the presence of another allele. So if you imagine there's an allele here, for example, that controls controls one aspect of phenotype, and then there's another gene over here that controls a different aspect. When you have 2 of them together, you get a sort of an effect that isn't, you wouldn't get when you have either of them on their own. So there are interactions between genes, oftentimes genes located a long distance away, maybe even on a separate chromosome. And of course, these aren't inherited together. Your odds of, if you're talking about 2 loci that are really close to each other, like, right here, the odds of you inheriting them both together are pretty high due to linkage. But the odds of you inheriting this locus has basically no relationship with the odds of you inheriting this locus. They're too distant. Anyway, so additive effects are the ones that we really care about in this context. And they're fairly simple to measure in a very simplistic way. They can be measured quite easily. What you really just have to do is measure is to correlate the trait measures either in parents or offspring or in MZ or DZ twins. Really anybody where you know the rough amount of genetic relationship they have. MZ twins, of course, are identical. DZ twins have half their DNA in common, etc. Wherever you have a known relationship between a pair of people, you can measure the correlation that they would have on a given trait. So here are some examples. This would be one with a fairly low additive heritability. The value that the parent has of this phenotype, these are scaled, Z-score type things, but the value the parent has is not very strongly correlated with the value that the offspring has. So there's no real relationship here. H squared is 0.2. On the other hand, if there's a positive correlation where the score that the parent has is very predictive or very closely related to the score that the offspring has, and you have a positive correlation here, your heritability is rather high, H squared of 0.8. So you can actually have heritability that's perfect, but still not have a perfect correlation between traits. But because the correlation coefficient here, which would be describe the actual strength of the relationship, is the square root of 1/2 H2. So even if you had an H squared of 0.9, the correlation coefficient would only be 0.64. So you're not guaranteed to have a complete perfect match. And I mean, the other thing to think about, by the way, is that we've already talked about how intelligence is heritable, but the heritability of it varies with age. The heritability score, you would measure it to be higher if you looked at older children. And heritability of height is also very high. The H squared there is 0.8, so very, very heritable. But at the same time, there's been a secular increase in height over the last century. People are getting taller and taller as time goes on. And clearly the genetics aren't changing. Yet somehow it seems like the height is going up. The same would be true for BMI and obesity too, by the way. I mean, BMI and obesity are very genetic, yet the rates of obesity are going up every year. A large heritability, by the way, also does not indicate how polygenic the trait is. That's a separate question. So you can have very heritable phenotypes, but they may involve many different genes. So it gets to be pretty complicated when you actually put this into practice. But anyway, that's all so that's a kind of just a very rough understanding of what I mean by heritability. But when we're applying that to this study, we can look at cohort studies or family studies like the Framingham Heart Study, which is a study out of the Massachusetts area of many, many thousands of families that all have kind of a known family lineage and a known family tree that connects each other. And it's possible by measuring brain volume in them to estimate the narrow sense heritability. So here from this study are three-dimensional reconstructions of various brain regions, and then estimates of the heritability. So these are the 95% confidence intervals. So for example, the caudate nucleus, the heritability is, or the narrow sense heritability, I should say, is estimated to be between 0.71 and 0.85. Brainstem between 0.79, 0.86, and so on for all these other regions. And I think what I found interesting about this is that the narrow sense heritability for amygdala volume is the lowest out of all of these subcortical structures studied. The heritability is as low, or could be as low as 0.34 or as high as 0.59. So certainly the lowest out of all of these. And I'll invite you to just reflect on this, why it would be that a place like the amygdala, the size of your amygdala has less to do with your genetics than the size of other brain regions. Think about, you just kind of sort of chew on that for a little while and thinking about what the amygdala does and how it's affected by various things. But the point I guess to make here is that surprisingly, the volume of subcortical brain regions is all fairly heritable. I mean, 0.86, that's a very, very, very high narrow sense heritability. That's approaching the level of heritability of things like height, which is sort of a classically well-known heritable trait. So at least when it comes to subcortical brain regions, the genetic contribution seems to be fairly large. And so they did a GWAS study linking the subcortical volume to different genetic variants. This is your classic GWAS study. We'll be seeing many, many of these. The classic Manhattan plot, we've gone over this. The y-axis refers to the negative log 10 transformed p-value. So the higher the number, the more significant it is. The higher the negative log 10, the lower the number, the lower the p-value is, kind of thing. The SNPs are given here. The lead SNP is named. You get the ref SNP ID, the RS ID. So you folks already know how to deal with those RS IDs. You could go look any of these up if you liked. The cutoff for genome-wide significance is given here. So any SNP that's above this line is considered to be significant, and so on. So the authors overall found a total of 48 SNPs that met the threshold for genome-wide significance, meaning that there's a total of as far as they know, there's 48 SNPs that contribute to variations in subcortical brain structure. The regions, by the way, are colored by what part of the brain they influence. So red, for example, is the brain stem, and so on and so forth. So genetic variants, or subcortical brain volume, pretty genetic. So let's look at another kind of technical note here and ask, where in the genome are these SNPs located? One thing you can look at when you see a Manhattan plot, by the way, is the points tend to cluster into these kind of little peaks. This is why it's called a Manhattan plot, is that there's like these little skyscrapers of points where all of the SNPs in that area are all kind of more significant than the surrounding area. You'll kind of have these like clusters or like locations where there's a lot of significant SNPs. They may not, surpass the genome-wide threshold, but they're all kind of nearby. And the thinking there, well, partly that's due to linkage, and we'll talk about that in a future lecture. But the thinking there is that these SNPs aren't scattered randomly. They exist in certain contexts that have some kind of regulatory importance in the way genes are regulated. So the challenge is to figure out where in the genome, like, what is the kind of regulatory context that these SNPs exist in? What are the epigenetics of the regions? If a SNP lands in an exon, it's easy to tell what's going on. It's going to be a coding variant, most likely, or maybe a synonymous mutation or something like that. But if it's in an exon, you can kind of work with that. But a very small percentage, as we know, are actually found in exons. So thanks to the work of epigenomics and gene annotation and all this other kind of stuff, we actually have a great deal of functional annotation for the genome. So for any given place in the genome, we actually know a lot about what's happening there. Think about your genome browser assignments. I would point you to a certain part of the genome and you would see all the different genes that are there, all the different expression data, all the different SNPs, all of the conservation data. There's also layers and layers of epigenetic data that can be filled in there as well. So for any given place, you know quite a lot about it. It used to be the case that these intergenic places between genes, these intergenic parts of the genome were described as junk DNA. They had no obvious function. They didn't seem to code for genes, and then what could they possibly be doing? But we now know that these areas have an important regulatory capacity or an important regulatory function. So what researchers are doing now is after they do a GWAS study, they'll try to see if the SNPs they've discovered are enriched in any of these regulatory regions. So let me give you a description of how that works, or one of the ways that this is calculated. So think back to your genome browser assignment and how you could find out where there were genes and where there weren't genes and about the overall sort of length of the genome and all this kind of stuff. Genetic and epigenetic features are described in terms of intervals. So an interval is nothing more than a chromosome, a start, and an end point. So you guys, or you folks rather, were working with intervals in this assignment when I gave you, CHR1 colon 1 million to 1,500,000. That's an interval. It just describes a little section of the genome on a certain chromosome. So when you're looking at epigenetic data, what you have are basically a stack of intervals where you might have hundreds or thousands or millions of different intervals talking about different places in the genome where that feature exists. So a feature could be something like H3K4 trimethylation, or it could be differentially methylated regions, or it could be ATAC-seq peaks. It could be a lot of different things. It doesn't really matter what it is, but it's some kind of data that you have. It could even be SNPs, frankly. And so anyway, now these are going to be positioned at various places in the genome. And so that's what we have here. So we have a little illustration of the genome, and there's two genes here, both pointed the same way. And then we have our H3K4ME3 peaks that are located here, here, and here. Now, these would be an epigenetic feature that you think is relevant. This is an epigenetic feature that controls some aspect of gene expression, so it's important. Now, if you have a second set of features, for example, SNPs that we discovered, you can test to see if they overlap the epigenetic feature of interest. So when it comes to your SNPs are located in points. They exist in one nucleotide. So they have a location that's not quite an interval. It's more of a point. But they can either overlap your H3K4ME3 peaks, or they could not overlap. So if a SNP exists within the interval, it's an overlap. And if a SNP exists outside the interval, it's a non-overlap. And you're asking statistically, do the SNPs overlap this feature at all, or do they not? So you have to kind of figure, like, how likely is it that if you just picked a bunch of random SNPs, what is the likelihood that they would overlap? You picked your SNPs because you did a GWAS study, and you think, therefore, that these SNPs are biologically meaningful. However, you don't really know that for sure until you compare them to randomly chosen SNPs. This would be like a null distribution that you might hear about in statistics. So what you can do is count the number of times you have an instance of overlap in your test set. So this is the set with the real data. So here we've measured 3 SNPs. This is a cartoon version. So we've measured 3 SNPs, and 2 of the 3 of them overlap with this epigenetic feature. One of them doesn't, and there's four instances of this epigenetic feature. So 2/3 of the SNPs overlap. Now, when you consider the overall amount of space that's available here, this is the kind of the whole universe of the genome. The odds are actually pretty good that a SNP wouldn't overlap, because if you're just picking random SNPs, they could go in any of these empty spaces and not overlap the epigenetic feature. So to test this, you can just generate computationally a random set of SNPs. So here is a randomly selected SNP. It's in gray, and one went there, and then one ended up there, and then one ended up there. So in this random set that was generated with no biology whatsoever, just you pick random SNPs out of a hat, the same number, but you pick the same number of random SNPs out of a hat, 1 out of the 3 overlaps. You can kind of do this many hundreds of times and get a null distribution of how many times would you expect SNPs to overlap your epigenetic feature by chance. Because again, random sets indicate the chance distribution, or what you would expect if things were happening randomly. And so in this context, you can measure enrichment by looking at the number of overlaps between SNPs in your test set and the epigenetic feature you're curious about, H3K, whatever it was, H3K4 trimethylation. You can take the actual number of overlaps and divide it by the average number of overlaps you get in a randomly generated set. So if you generate 100 random sets, maybe the average overlap you get is 1/3, or 0.33%. But in your real data set, you got 2/3 overlap, so 0.66%. That means that your test SNPs are twofold enriched in these H3K4ME3 peaks. Does that make sense? Anytime you're talking about enrichment, you're basically trying to make a comparison between what you got, like what the actual overlap is between a set, and what you would expect if you were giving it random data chosen out of a hat that's the same size. That's the essence of this test. And it is quite similar in logic to the way the gene ontology enrichment works, which, again, the assignment is now available. You can go and do that at your leisure. So I wanted to kind of, take a moment to describe that in enough detail that I hope would make sense to everybody, because this concept or this motif appears fairly often. We have all this data on the epigenetics of different parts of the genome, and we have all this data about SNPs and about other features, and we want to know, is there any kind of overlap between them that would tell us something about what's happening at these locations. So here, the authors of this paper looked at the enrichment of the SNPs they found, the 48 SNPs they found, and various functional annotations for the genome. So this is really quite an ugly plot and a little hard to read. So we'll just kind of highlight the different key elements of it. The enrichment score is given by the color scale. So red means highly enriched, 27-fold enrichment. Blue means depleted. So what that means is actually there's less overlap than you'd expect by chance. Sometimes you could have the same amount as you'd expect by chance. That would be an enrichment of 0. You could have way more than you expect by chance. That would be a positive enrichment. Or you could have way fewer overlaps than you'd expect by chance, and that would be a depletion. So you can go through and look at these different annotations. There's all kinds of ones here: 3' UTRs, coding sequences, highly conserved regions, CTCF binding sites, DNA's hypersensitivity regions, enhancers, H3K27A, and so on and so on and so on down the line. All these different things that exist in the genome. Many of these you'll recognize by the time we're finished, but not all of them. There's too many to go over here, but many of these you'll have some familiarity with. The overlap between these epigenetic annotations and the SNPs that are linked to brain variation in brain region size. So just to take a single example, we can see that the genes that are linked to, for example, the size of the brainstem, the amygdala, brainstem, all the way down to the putamen, are all enriched in H3K27 acetylation peaks. So places in the brain where there's a lot of H3K27 acetylation, that's a marker of enhancers, or active enhancers, I should say. Areas of the genome that have those in the brain tend to be enriched for SNPs that control the size of these brain regions. And same goes for super enhancers and a few other marks as well. So what this tells you is that that these enhancers that are marked by this H3K27 acetylation are important for brain development. So this is a way of linking the epigenetics, which is this histone modification, with the genetics of brain development in a way that may potentially affect the size of different subcortical brain regions. So again, this is not the kind of thing you would have to remember or memorize, but it's a concept that I'd like you to understand, is that you can measure the enrichment of SNP, of phenotype or disease-associated SNPs in different epigenetic annotations, and that gives you a meaningful piece of information. So there turns out that there's a surprising amount of overlap in the genetic variance that determines subcortical volume. And this kind of makes sense because brain development is centrally regulated by a handful of really quite important transcription factors and morphogens. And regions that are anatomically and developmentally related tend to share more of these candidate genes. So for example, the caudate, the globus pallidus, and the putamen, all parts of the basal ganglia, all have the same genes that influence their size, which is, I think, kind of interesting in its own way, because neuroanatomists, centuries ago figured out that these parts of the brain were linked to, were kind of the same, or part of the same overall structure. They defined the basal ganglia quite a long time ago, long before any of this was known. And it turns out that when you look at the genetics and the epigenetics of brain development, they match very well with the work done by those early anatomists who were literally just looking at, brains under the microscope and trying to think about things and just thinking about things very carefully with what they could observe using their eyes. And in many cases, they discovered knowledge that's equally as good today as it was back then. And how many regions, or how many parts of science do you know where work that was done over 100 years ago is as true and valuable today as it was that long ago? And I would say probably physics, maybe a little bit, but a lot of areas don't work that way. So big shout outs to the big shout outs to the early anatomists for doing such great work. You really have to hand it to them, folks. You really got to give it to them. But anyway, so the variants that they discovered, talking about the genes themselves, there's a wide range of things, one of them being IGF signaling. IGF-1 itself has an important variant that's linked to subcortical brain structure, but the IGF axis, the insulin-like growth factor axis, is actually fairly well known for its role in somatic growth. IGF-1 is released in response to growth hormone, and that's what actually causes a lot of the effects of growth hormone on your somatic growth. There's other genes involved in autophagy and apoptosis, inflammation, and immunity. There's a whole host of genes, but what I'm kind of trying to show you here is the GWAS model can be applied to a very wide range of traits. Now, I've got bad news, folks. Phrenology is back. We're back in the game of measuring skull thicknesses and shapes and whatnot. Not really, but I had to bring it up. People have really thought for quite a long time that brain shape conveys some kind of insight into somebody's personality or their intelligence or the behavior. This is very, very old. There was, the ancient Greeks had a concept called physiognomy that was a way of assessing personality from facial appearance. Of course, there was various cultures have had versions of this as well. There was sort of a Chinese tradition called face reading that had a similar kind of idea to it, and countless others. Phrenology itself was sort of a revival of physiognomy popularized by Franz Joseph Gall, who, basically any theory from those days, it was really, any theory from any day really was quite rooted in racism and sexism, mostly just justifying eugenic theories. So the idea was that, there's a reason us Europeans are on top. It's because our skulls have the right shape, all this kind of nonsense. But it was quite popular for a time before it was debunked and rightfully consigned to the gutter of pseudoscience. I like this picture here. This is an example from a physiognomy textbook where they're trying to argue that personality traits have to do with your head shape. And I thought it was just funny because it was sort of like, this would be over 100 years, this book, but it's sort of like an early version of that, that Virgin Chad meme. But anyway, this is very commonly held thought. Even if you know it's fake, you still can't help but read people's faces a little bit. And researchers do study physiognomy. There is data even to this day that show that as far as men's facial width to height ratio, there is a correlation there with aggressive tendencies. And there have been many sort of ill-fated, ill-informed attempts to make this more scientific by applying machine learning and whatnot. So it's kind of a controversial area, but suffice it to say, face shape does sort of mean something, maybe. Brain and face development are integrated. This is what's surprising about it, but not as surprising as you might think. The facial skeleton is very close to the developing brain, and in fact, the facial skeleton actually comes from the rostral neural crest. The neural crest cells are what actually give rise embryonically to the face skeleton. And as the face is developing, it's right next to the developing brain. And in fact, the developing face seems to react to the physical growth of the brain. This is something they've discovered a lot in chick embryos, for example. They once did an experiment where researchers transplanted an embryonic duck's brain into a chick, and of course the duck's brain grows faster than the chick's brain, and as a result, the brain or the facial development of the chick was delayed. The brain kind of orchestrates facial development because the brain is the most important thing in the head, and it sort of gets the first say on how things are going to look. So there's bidirectional signaling between the processes. And people have looked at this also in mice. There was a study back in 2008 where they selected mice for short faces. You can selectively breed mice to have shorter and shorter faces, kind of a more neotenous appearance, really. And what the authors of Bogner et al. Found was that short-faced mice also have smaller brains and overall slower brain development. But during the fetal stage, because brain development was slower, facial growth was actually advanced in these mice. So facial growth in these embryonic mice happened ahead of schedule. But it also ended ahead of schedule. So it started ahead of schedule and it ended ahead of schedule, ultimately not reaching the same length that it would normally reach. So it was shorter than normal. So what they're kind of saying basically here is a mouse that was selected to have a short face, what was actually happening was it was selected to have a smaller brain. And the smaller brain is what actually caused the mouse to have a shorter face by making by making the face development start earlier and end earlier, and thus not manage to get as long. So kind of a neat concept, and so there is this connection there that is maybe not very well known or appreciated, but does appear to be true to a certain extent. And there are growth factors like NKX2.1 and Sonic Hedgehog from the developing forebrain that influence face development directly. There's only so many growth factors in the body, right? There's only, dozens or a few dozen or so different growth factors that are even available. So if you have two things growing right next to each other, it's sort of bound to be some influence between the two, especially if they use similar signaling pathways. Anyway, this year, NACV and company we're basically trying to study the genetic influences on brain shape. So this is another example of the genomics applied to the world of neuroanatomy. So the way this works basically is when you conduct an MRI scan of somebody's brain, you take their data and you can convert it to a three-dimensional model. It's what's called a mesh. A mesh is a 3D model because it's a mesh of vertices floating in three-dimensional space. These can be fitted to kind of atlases that convert all of the points to kind of a normalized space. So you can scan hundreds of different brains and always find out where the hippocampus is because the points have been kind of normalized to a prototypical brain. You can also hierarchically segment the brain development and divide the regions into smaller and smaller subcomponents. So this is an example of how this is done. This is a very multi-dimensional form of data. So on the first round of segmentation, they break the brain down into two regions, anterior basically and posterior, or rostral and caudal. Then those are broken down subsequently into smaller subregions, and then smaller and smaller subregions, and so forth. So there's this hierarchical relationship between the bigger regions and the smaller regions. The smaller regions are a subset of bigger regions. So this is a circular dendrogram. It's sort of like one of those family tree dendrograms that we've looked at, and it shows how the brain is first divided into the brain, and then it's divided into two rostral and caudal, you could say. And then each of those sets of lobes are divided into smaller and smaller substructures until you've broken it down to very, very small brain regions that all can be distinctly defined. And the beauty of this is it converts this sort of three-dimensional data into a sort of a numerical multi-dimensional data set that you can use with traditional methods like principal component analysis and so forth. Let me just pause. I got a question here. So Alexis asked, let's say a developing fetus was expressing trisomy 21, Down syndrome. That would play a role in the development of their facial features? Absolutely, yes. Trisomy 21 certainly has a facial phenotype that's quite characteristic. Although I wouldn't say, off the top of my head, I don't know if this is because, if it's due to the relationship between the developing brain and the face, in the way that I've described, or if it's some other separate thing where having an extra 21st chromosome causes a variety of, abnormal phenotypes, and one of them just happens to be the face for some other reason. So I can't say for sure that the facial structure that's characteristic of trisomy 21 is because of this relationship between brain development and face development, or if it's because of some kind of other effect it's just related to the 21st chromosome. Interesting question, though. Yeah, it's very curious to ask. Because that is one classic example of a link between the two. So you've got this kind of multi-dimensional data set that's been clustered or been kind of decomposed from the three-dimensional brain. And I wanted to point out that this is not something only for neuroscience. This is called hierarchical segmentation. And it's actually a pretty important part of deep learning and AI in general. Any three-dimensional mesh, any mesh could be subjected to the same process. It doesn't have anything really to do with the brain. It's just that you could apply it to the brain and get kind of unsupervised segregation of the brain parts, which is nice. So here's an example of this from a paper done in 2017 showing how it can be applied to 3D models. So here's a car, and you can break the car down into different subcomponents, for example, the doors. And the doors can then be broken down into further subcomponents like the windows and the rocker panels and so forth, and all the different subparts. And wheels, of course, could be broken down into tires and rims and all the rest of it. And so this is a concept that applies to three-dimensional meshes in general. And if you take this approach that could be used in AI to help recognize objects, you can also apply it to MRI-derived 3D models of the brain and break the brain down into subcomponents. And this is just kind of nice because it's automated. You don't have to sit there and do it yourself for all the thousands of different brain MRI scans you have. And it's also it's agnostic to our own anatomy, our own preconceived notions of anatomy. We're not telling it where we think the subiculum is, it's going to find whatever it is that varies between people, and regardless of what we've named it. So this is a very complicated picture, but it's just showing, it's sort of a karyotype of all the chromosomes, and it's showing regions on the chromosomes that influence the development of different parts of the brain. So you've got white icons that indicate places that influence the development or the shape of the whole brain, and then other ones that are broken down by which quadrant they influence, or like which section of the brain they influence. So you can see that the genetic influences on brain shape are spread quite widely throughout the genome. I don't know why they didn't just make a Manhattan plot, but anyway. Oh, here's the Manhattan plot. So here's another view of this, where they're showing, as with any Manhattan plot, they're showing the significance of the association between a given SNP and the shape of a certain part of the brain. Here they've kind of collapsed all the shapes together. They're not looking at certain regions, they're just looking at any SNP that has any link to brain shape whatsoever. And so there's a couple ones on here that make a lot of sense. DLX5 and 6 are transcription factors involved in brain development, the bone morphogenetic proteins, and so on. And you might notice that this is a little bit different than a regular Manhattan plot, because there's also an underside to it. This would actually make it a Miami plot. Miami plots are like Manhattan plots, except they have like a reflection. You know, Miami is on the water, so you can have like the skyscrapers are like reflected in the water. So here they've plugged in a Manhattan, or sorry, you know, a plot showing the significant, or the relationship between significant SNPs controlling face shape, and they've just superimposed it on the SNPs they found controlling brain shape. And you can see that many of these areas are very well reflected. So this DLX5/6 SNP that's really associated with brain shape is also strongly associated with face shape. By the way, the face shape was determined using the same kind of 3D methods as the brain shape. So just looking at it, it seems like there's a fair bit of overlap between the genes that affect face shape and the genes that affect brain shape. Isn't that funny? So you can look at where these genes, or the genes linked to these variants influence. And so, for example, variations in PAX3 control the shape of sort of this anterior part of the brain, the frontal cortex mainly, and a little bit of the temporal lobe. But they also control the development of this part of the face, kind of the frontonasal area. And you can kind of go on down the line. This part of the brain, patched 1, or the patched 1 controls the development of the occipital lobe, but also seems to affect nose shape. So pretty weird and wild stuff if you think about it, that for these variants, the effect they have on the brain is fairly correlated with the effect that they have on facial development. So here's another way of looking at it. Here's the genetic correlation. So this is the Spearman correlation between variation in SNPs and the shape of the face and the brain. So for example, the variants that control the shape of the sort of the frontal ridge part of the face, whatever that's called, are well correlated with the shape of the frontal lobe, not this kind of parietal lobe area, or but more sort of the like the prefrontal cortex type area. So I'm gonna go down the line and see how the genetic variation in different brain regions, or in different genes, controls variation in face and brain in a kind of predictable way, which is a pretty strange discovery if you think about it. Now, it doesn't explain personality. So that's the next thing you'd want to ask, right? Like, now that we've got this sort of scientific version of phrenology, can we use it to like measure people's personality? Or could we look at somebody's face shape and predict whether they're going to be a good worker, or whether we should charge them more for health insurance or something like that? The answer is no, as far as they can tell. So, we wouldn't love it. Actually, it would be horrible if it were true, but it doesn't seem to be true. So you can look at the correlation between brain shape, or the genes that are controlling brain shape, and your odds of having, and the genes that relate to schizophrenia or bipolar disorder or ADHD. We'll talk about genetic correlation in a future class. It's a pretty important topic. But for now, we'll just say the genes that control brain shape are strongly correlated with the genes that control schizophrenia or ADHD or any of these other conditions, as well as the genes that control these personality types. So there is a relationship between the genes that influence brain development and the genes that influence various aspects of your personality and your risk of psychological disorders. So that's pretty important, because those conditions, personality and psychological disorders, are rooted in brain development. So there is an overlap there between the genes that regulate brain shape and the genes that regulate these conditions. However, there is no overlap between the genes that regulate face shape and any of these conditions. So based on that analysis, there's apparently no link between kind of the genetics of facial structure and the development of mental illness or personality. So I guess that's probably better news, I guess, for all of us. We don't have to worry about our faces getting scanned and having it linked to our personality in a really kind of predictive way. But it's a complicated topic, and it's one of those things where like it would almost be so bad if they found it was true that you wouldn't want to find it. So I just want to say, like, so the genes that are linked to brain and face shape tend to be transcription factors or morphogens. And as I said, there's only a handful of these even in the genome. So typically if you have a mutation in one of these or a variation in one of these, it's likely to affect a lot of different things in a sort of similar way. This would be called like a pleiotropic effect. This is when there are sort of like many correlations between a gene and phenotype all across the body, because like SOX9, for example, which is one of the genes they mentioned, is involved in neural crest development, but it's also involved in chondrogenesis, gliogenesis, and craniofacial morphogenesis. So it's kind of hard to say for sure what's really happening here. You could be looking at all kinds of parts of the genome and maybe finding correlations here and there. What it really means, who knows? It's important to point out that the study was done on white Europeans mostly. So whatever it means would only really apply to them. At a bare minimum, it would only apply to people of white European descent. And the fact that the brain and the face develop in such close proximity suggests that this is an example of like a paracrine mechanism where signaling molecules secreted by the brain kind of influence the development of the face just by virtue of their proximity. It's also an example of a pleiotropic effect when one gene can influence many different traits. And like when it comes to that, you're not going to do much better than these growth factors, because growth factors control all kinds of aspects of development in a very precise way. And I would say, as a final warning, please watch out for people who think they can derive meaningful brain information from the face. There does seem to be a relationship here, that's clearly the study is showing that, but it's mostly at the level of genetic correlation, which is a separate concept from like a real anatomical correlation. Genetic correlation is a very specific technical thing that what it means basically is you're not going to be able to just take somebody's face and be able to really infer their brain shape, at least not with the technology we have. So beware of that. We'll talk about genetic correlation in an upcoming class, so you'll hear more about it then. I want to just point out this is something I sort of found funny was like predictive algorithms are always out there trying to take one type of data and use it to predict another one, and they're only as good as the data you put in, I guess. But I thought this was kind of funny. Somebody put in a picture of somebody who's clearly Obama, and the predictive algorithm guessed that it looked like this guy. So it'll pick out features that are sort of similar, but it doesn't have the deeper contextual knowledge that we have. And generally, I guess when you're talking about the relationship between things that are really important to people, face shape and brain shape and intelligence and various personality characteristics, you have to be extra careful with the claims you make and with the way you drive your research, because maybe there is a relationship between brain shape and face shape and personality, but you'd want to be very certain of that before you started making claims, because there's a lot of societal consequences if such a thing were discovered. You can kind of imagine them yourself. So watch out for machine learning grifters. Watch out for people who are just trying to make cash by claiming they can do a certain thing when they really can't. Let me go to the chat here for a second. So Natalie asks, are the intergenic parts of DNA, i.e. junk DNA, the introns in the genome? I can't remember which slide this was on. Right, the junk DNA stuff was, where was that? Oh yeah, this was back on slide 18. So let's see here. We can go back to slide 18 here. Or no, not slide 18, slide 19. So junk DNA, I mean, introns would be one of the examples of what people used to call junk DNA, as well as intergenic spaces. So intergenic just means any area that's not inside of a gene. So introns are technically inside of a gene because they exist between the start site and the stop site for transcription. They're not transcribed, or they're not transcribed in the way that exons are, but they are present in the gene, so they'd be intragenic. So junk DNA would be anything that's either really just didn't have an official term, but it was anything that sort of couldn't be explained. So introns and intergenic spaces would be both part of that category. Yeah, and like I say, that's an old-fashioned term. Nobody really used it. And it's actually kind of a straw man too, because even when people were throwing that term around, geneticists and people really working in the field did not use it. People didn't really think that the genome could be full of junk. It doesn't make a lot of sense. Evolution doesn't tend to work that way. But yeah, so that's the answer to that. So it could be anywhere. But we don't believe in junk DNA in this class. All the DNA matters. It's all doing something most likely. All right, my friends. So that's it for today's class. Like I said, you can check out the second assignment when you get a chance. It's due in November 8th. And stay tuned for your quiz marks. I have to figure out how to upload those in a way that's not going to upset everyone. Well, I feel like when I'm handling marks, like I'm pretty comfortable with Brightspace at this point. But when it comes to handling marks, it feels like you're handling like nuclear spent fuel rods or something. You really have to be very careful, because if you put the wrong mark up, or if you erase people's marks, or if you do anything that makes weird things happen with the marks, that's big problems for you. You're not going to be happy, because students will see their grades swing back and forth if they see their grades go really low or really high. Or you have to just be very careful of this kind of stuff. And if you don't have the marks recorded elsewhere, you could be in big trouble if you lose them. So just bear with me while I get those set up and visible to everybody. And like I said, the office hours are tomorrow, and your TAs will be contacting you with information on how you can talk to them about the grades or about the marking that they got for their exam. So thanks to everyone for listening. I hope you have a great day, and we'll see you next time.